Well, hey there, my lovely IB Bio students. I am so sorry this is coming out a little bit late. I will try to get ahead with our lectures. Okay, so we're just going to go straight into it. So today we started looking at cells under microscopes. But remember I told you guys in this unit we're going to talk about cell theory and kind of move forward with new stuff. But also I want to go back and review some basics about cells that are kind of I'm going to say implied that you know in topic 1. And so we'll go back tomorrow also and look at like prokaryotic and eukaryotic and the different organelles as well, because I know you guys saw some of that today. All right, so topic 1.1 is our cell theory. Remember that in biology or sciences, theories are things that are not just guesses. Like I have a theory, they're backed up by a lot of data. They are scientific models, right? There's actually really 3 big scientific models in science, and that is the central theory, the central dogma, and natural selection, right? Okay, so in this section, you don't have to write this down, but by the end, if you want to go back to this, we are going to be looking at what are the functions of life, right? How do we even know if something's living? What is the cell theory? We're going to look at how it's changed over time in class. We've already started microscopes, so you don't need to do that. I'll skip over that part. We started magnification, which you guys were super excited about, but I'll have you guys, if you did not write it down in class, write that down. We'll talk about why cells are so small, what multicellular organisms are, how they become different, and then stem cells. I love talking about stem cells because that's like a kind of hot topic right now in science, and there's a lot of fun research that you guys can find on that. Okay, I feel like this is cutting. Hold on, is this cutting it off? Oh no. Okay. Let's see. Let me see if I can get that to not cut off. Okay. All right. Let's go here. Okay. So functions of life, right? So the 7 basic. You might have. I just said basic because I was going to say basic functions. You might remember learning this in as the 7 characteristics of life. This is basically when I look at it, is like how we like. What do I want to say? Like outline our year, right? Like all these different things. But the 7 basic life functions, and this is definitely something that can be testable. You can remember with this kind of this acronym, right? Miss-Hang. All right. And so what those stand for, and we'll just write it down for now, but know that like we do an entire unit on most of these things, sensitivity a little bit. But anyways, the first one's going to be a metabolism, right? So you have probably heard about that word metabolism as in only pertaining to food, right? They have a fast metabolism or a slow metabolism. But metabolism is just the speed of chemical reactions, right? But all living things have a metabolism. They go undertake these essential chemical reactions that break down things that you put into your body, whether that's air, food, things like that. Or they and then they make it into something else, right? So metabolism is that chemical reactions that occur. So we know that all living things have a metabolism. Hold on just a second. There we go. Okay, we are just going to keep it like this. You can see all my tabs open, so you can see the only thing. Okay, let's see. Oh no, stop it. Okay, so metabolism, first basic function. Second one is that it has the ability to reproduce, right? So the ability to reproduce. There's a couple exceptions to these. I should talk about that. Like in biology, we always talk about they produce offspring. So we talked about some mixes, like hybrid mixes, like a lion and a tiger. Well, they can create an offspring, but that offspring can't create offspring, right? Because of the abnormal chromosomes. So does that mean that it's not living? No, it still does, right? But that it has the ability to reproduce, that it creates, sex cells. If it's eukaryotic, right? Or if not, if we're looking at bacteria, we're talking about asexual reproduction. Again, kind of a brief reminder from bio that sexual reproduction is when we have 2 genes coming together, right? So you got sperm, egg coming together to create a new mix of things. Asexual is just when it is able to replicate itself. So we see asexual reproduction in like bacteria, right? Things that are just able to replicate themselves. All right, responding to the outside environment and internal environment. This is called, we call this sensitivity, and it links right in here with homeostasis, right? So sensitivity, that characteristic just means like things change on the outside and there's a response inside. Things change on the inside and there's a response outside. For example, things like plants, right? So you can think of like humans, a lot of examples, but things like plants, if there is external stimuli like a light changing, right? Like they're changing its environment, it responds to that, right? So we don't see it like just maintaining. Now it responds to it going into that by homeostasis. That is the process by which it maintains a stable internal environment, right? So your temperature heats up, right? Your homeostasis, there's things that are in your body, mechanisms working to bring it back down, right? You have too much, of a chemical in your body. It's going to metabolize it back out so that it comes down to where it needs to be. Anything like that, right? Like low blood sugar, high blood sugar, any of those examples. All right, so sensitivity leading in homeostasis. The E stands for excretion. That means it has the ability to remove toxic waste products. Lots of different examples there we can talk about throughout the year. But with humans, you actually don't do anatomy physiology for IV bio until year 2. But with plants, being able to excrete toxic waste like carbon dioxide, right? We excrete carbon dioxide, right? But getting rid of those things like excess nitrogen, etc. Nutrition, so eating, exchanging materials with the environment. Again, so bringing in food, breaking it down, making it something else. Our exchanges that we give it back carbon dioxide, right? So we break down the carbohydrates and give that back. Plants do the opposite, but that is classified as nutrition. And all living things have the ability to grow or move, right? So not plants don't always move, but we also call that growth as far as like change shape, size, and positions, okay? And this is Mr. Oh, that's so funny. I was always taught it as Mrs. Hang, but this is Mr. Shang. Look at that, 2 mnemonic devices there for you. Okay, all right. So let's go into, remember your DBQ on paramecium. All right. You do not need to write this down, but this is just kind of an example of those different things. Again, Mrs. Hang or Mr. Shang, but looking at like what type of metabolism do they have? Are they autotrophic, making their own food, versus heterotrophic, right? They need food from the outside. How do they reproduce? Asexual, looking at how they are sensitive, right? So what they have those trophisms to, how they maintain equilibrium, how they excrete, right? So for diffusion versus pores, how they actually get the food in, right? So these are different like functions, of those functions of life into different types of cells here. Paramecium, the one-celled heterotrophic, that means food, versus like this is the plant cell here. Okay. All right, so here is the cell theory. In class, we're going to watch this short video on the wacky history. I was telling you guys how much I really love like the history of science. It was a fun class. All right, so the cell theory is actually, you need to know these like 3 main points of the cell theory. Pretty basic. My son just learned these in 6th grade. I was actually really impressed. Okay, this is how we structure and organize all living things are based on these 3 assumptions, part of the theory. All living things are composed of cells, right? All living things are composed of cells or cellular products. We'll talk about that kind of exception. They are the smallest unit of life, and all cells come from pre-existing cells, okay? Composed of cells, smallest unit of life, all of them come from pre-existing. Now, there's some problems with those we'll talk about, but those are how, like the 3 big ideas that make up the cell theory, okay? Now, here are some different exceptions. So you can kind of used to like the IB test from now is like you do need to know exceptions, right? So and even examples, right? So now there are certain types of tissues and cells that we would still call a cell, but don't always conform to that definition of a cell. Okay, so those 3 examples, and I think I have slides from this. I'm pretty sure I do, so we can look at them in class. But striated muscles. So striated muscles are the muscles, like instead of having like the circular, they're really long, and they fuse together to make these they're called multinucleated fibers. So you have all these different nucleuses, all fused together in these long patterns to create like this complex though, but they're all together and they share the same nucleuses. Okay. The giant algae is another kind of like outside the standard of what a cell is. They can be very large, right? So you got very large types of algae. So when we talk about cell size, those will come back to that. And certain types of fungus, right? They can be connected through cytoplasm. And again, we can look at those in cells, but you should know those cell theory exceptions. So there's like 3 specific types, might not always conform to those notions of what a cell is. Okay. All right, cell scale. So we talked about this just a little bit in class today with your calculations, all right? But if you did not, so pause, if you did not write this down in your notes on calculating magnification, let's make sure that we have that in there so that you know the difference between the meter, the millimeter, the micrometer, the nanometer, and the picometer. Now, today we only saw these 3. No, we just really saw these 2, right? The millimeters and the micrometers. But we do need to know, so I think it was notated like this in your book, but make sure you have this written down so that you know how to go from one to the other, right? So if I have a millimeter, I can divide it by, this is divided by 1,000, right? So when we did today, we multiply it by 1,000, right? So that we have 1,000 millimeters, right? And a meter. We have 1,000 millimeters and a micrometer. So we can go through those ways of looking at the magnification. Okay, so our cells were usually measured in micrometers or nanometers. Again, we looked at the micro today. We did not look at the nano. But here's some examples of how big these things are. And so you can see like molecules, right, being looking at the smallest forms here, membranes, right? So they're kind of going up in size as we look versus the cell being the biggest thing, the things that like organelles are smaller within them. Okay. All right, microscope. So you already wrote this down today, the light microscope, but you should know that like being able to tell the strengths and differences between light and electron microscopy. So we do not have electron microscopes, but you will be looking at light. And just so you know, like outline those 2 different differences, right? So these are things that we cannot see with the naked eye, right? We need some type of magnification. The light that you guys looked at today looks at them in their natural color, if they have it. We'll talk about the dyes, and has a lower resolution and magnification. Electron microscopes with way more detail. They look in monochrome, so you do not see them in their color. They have this like grayish right here, but it has a much higher resolution and magnification. So here's 2 of the same ant here. Here's the light, and here's the electron, right? So know the differences between those 2. Here are some examples of some light microscopes. You do not need to draw these down. They'll be in your book, but just so you know, like even look at the sizes, the size of the bacteria versus the plant cells versus animal cells and muscle tissue. You will be able to have to differentiate between plant and animal cells. I talked about that a little bit today, but we'll look at like the different features of the plant cells, like the cell wall, the chloroplast, the things that are like features so that you know that it is a plant. Okay, here's an electron microscope again. You see these differences in their resolution. You can see the structures a lot better. And this is one that color has been added to. So it's telling you the difference between here's how big a cell is, right? Here's the bacteria on it, and here's what a virus is. So you can see that kind of scope of how big those things are. Okay, again, if you wrote this down in class, do not write this down again. Okay? If you didn't have these things mixed around, I think 3rd period, you wrote it down. I don't know if 1st period I made you write it down. So make sure you have these on that white piece of paper where we drew the microscopes. Okay. I like those kind of mnemonic devices of MIA or AIM, right? So how do you find actual size versus magnifications? We'll do more practice of this in class. I'm actually going to go to the bank and see if I can find more questions than the one in your textbook. Okay. Yep, just going to skip this one. We went over in class. If you didn't, I'll put it back up for you guys tomorrow. Okay, so magnification, now going into surface area. So really 2 different things. So we're kind of transitioning. Okay, so the other thing that's important to know about cells is their surface area to volume ratio. I have a demonstration for you guys this in class, and it's super simple. So we'll kind of go through it. But basically, cells need to be small so that they can easily exchange materials with the things around it. Like that's the basic of it, right? The rate at which they can exchange is going to determine how fast they can move things in and out, and therefore like keep their cell healthy, right? So the rate of metabolism is a function of cell volume. If you have a large cell, it's going to take longer for things to break down and be shipped out than it is if it's smaller. Same thing with heat exchange, right? Things that are really big versus really small. If I need to get heat out, it's harder if they're bigger. Okay, so as the cell grows in size, the volume will increase faster than its surface area. Let's say that again. The volume to surface area ratio, the volume gets bigger than its surface area, increases faster. So this leads to a decreased surface area. So my volume ratio, right, as that decreases, you guys can write that down right to the volume. Okay, so as cells grow in size, the volume increases faster than the surface area. Okay. In metabolic rate, if it exceeds the capacity, if it can't exchange, so why that's important, if it can't exchange at the rate that it needs to, it will die, right? It can't get the things in, it can't get the things out. You're going to see cell atrophy, which just means it's no more cells, right? It dies off. So growing cells will divide before they get too big, right? So we'll see like mitotic cells, like they grow, they pinch in half, and they go away, right? Not go away, I'm sorry, they separate. So I see cells grow big before they form, and then they pinch off to become two, because in order to survive, they have to cell small. They have to stay small. Okay, we'll look at some examples here. So if you look at, here is the ratio. So again, you're looking at the total surface area, the total volume, and then the ratio of surface area to volume, right? So if I have this here, and here's my total surface area, here's the volume, so the ratio is 1.2. As that gets bigger, so the surface area increases, but the volume stays constant, right? I see the surface area increase here, my volume stays here, so now my ratio is 6. Right? So as the surface area increases, the volume does not as fast, right? We don't see it go as fast. Cells do increase their surface area to transfer things faster, right? So if I need to get things out faster, I can increase my surface area so that I have more ability to do those exchanges, right? So versus if it's in the middle, right, and I don't have a lot of surface area, it has to move all the way out to the surface to be able to do that exchange, okay? All right, multicellular organisms, what you need to know about, like, so we have these single cells, but how do we group these together to become different, like to become multicellular, very complex? Should be review. We're going to go back through it. But multicellular organisms are when a bunch of different individual cells come together to do certain jobs, to all function together to make a living thing, right? So all these different cells, though, have different functions, so very specific jobs within the body or the organism that all work together for the common good of staying alive, okay? And so, like, you have cells, you do need to know the hierarchy of these things, right? Cells, common cells come together to make tissues, tissues come together to make organs, organs come together to make systems, right? So that's like the hierarchy of how things come together. So this example is like, I might have muscle cells, those come together, a bunch of cells, muscle cells together are called muscle tissue. If I have different tissues working together, they come together to make an organ like the heart, right? So I've got muscle cell here, but I also have my, I've got my blood cells in here. I've got all these other different types of cells in here working together and tissues working together to make the heart. And then that organ is part of like the cardiovascular system. So the heart works with the lungs, it works with the vesicle, right? So all those things that come together to work as a system. Okay, you can do that through any other things, right? We can have, stomach cells that come together and make the stomach lining tissue that works with the stomach, right? That goes to the digestive system. So that is the hierarchy of the order of multicellular organisms. Okay, cell differentiation. So how does that work? If all cells come from pre-existing cells, right? There's like the kind of like the crux of it. So how does that work then? If I have one cell, sperm meets egg, right? And then all of a sudden it divides and it divides and it divides. And so if all cells come from pre-existing cells and they're just multiplying, then how do I suddenly get all these different types of cells? How do I get brain cells, right? And blood cells and tissue, like how does that all happen? Skin cells, if they all came from the same thing. This is through a process called cell differentiation. And it's basically this kind of like chemical system that when all of your DNA has all of the directions to make everything, but only certain parts are expressed depending on the environment that they're in or the chemical messengers that they receive. right? And so here's the example, I've got these two identical cells, all with the same DNA, but in this cell, gene A is expressed, and in this cell, B is expressed, right? So I get the difference, like neuron in the skin cell, right? So the directions are all the same. You can almost think like it's a big book, and I said, okay, now turn to page, 42 and copy that. Well, page 38 is still there, but I'm not doing anything with it, right? And so another cell like turns to a different page. So all the directions in every cell, but that is the process by which I have one cell becoming different cells, cell differentiation. So those directions in the genes, I kind of just went through all that, right? Directions in the genes make it become something different. Okay. The activation of those, which cause those to become, is called specialize or specialization. And we'll talk a little bit about chemical and hormone and specific messengers that create that or like make that happen. Okay. Now all of these come from, I skipped this real quick. So gene packaging we talked about a little bit inactive genes in our last unit, but we have within our nucleus, we'll go back through organelles, but remember that our DNA is stored in eukaryotic organisms inside the nucleus. That's that super protective area around, right, that holds it together so nothing can mess with our DNA. Okay. Within the nucleus, we have active genes, and they are the ones, so within a cell that's specializing, they are the ones that are expanded, aren't accessible for the chromatin, the euchromatin to come in and make the protein that comes with it. Okay. The inactive genes, the ones that I'm not using, are packaged and condensed in the heterochromatin. Right? So the euchromatin, it's almost like, okay, copy this really quick, right? And all this other stuff, it just kind of stays packaged and condensed in those cells, right? And then in other cells, right, so we talked about gene A versus group B, like other things are exposed, and that's the euchromatin, and the heterochromatin is all condensed. So cells will differentiate based on whatever portion of the DNA is being, you think of like uncurled at that time to have that read to make into proteins, okay? All right, now stem cells, this is the last part. So stem cells are the cells in your body that are able to be made into anything, right? So stem cells are unspecialized and they're not really like determined to be anything, but they have the capacity to specialize into other things. So the cool thing about stem cells is they self-renew, they'll continually divide and replicate. They're only in a few different places in our body. I'll go that in a second. And their potencies, they have the capacity to be any type of cell. So you might have heard of like stem cell therapy, where it's like they take that, and so if you in the umbilical cord, and they'll keep that, and so that if later in life, like you need a specific type of tissue, your stem cells have that capacity to become whatever they need to be. Stem cell therapy is being able to use those, like it's been used in like it's experimental. I'm going to say like it's still kind of being, it's definitely in the stages of testing, but have had positive things with like skin grafts and stem cells, different types of like tissues being grown. But anyway, so embryonic, right? So stem cells that they find in the umbilical cord, those are called pluripotent or totipotent, which means they can form any type of cell, right? Adult stem cells have a limited capacity for differentiation. So the embryonic cells are different from the adult cells. You should know the difference between those two. So the pluripotent or totipotent, that means that they can form any type of cell. The stem cells in adults are multipotent or unipotent. So they have a limited capacity, but can still make different things, just not all things. Okay, so this is what I was just talking about with stem cell therapy. Replace disease damage, they're extracted, they're put in a certain solution where they can differentiate, they're then implanted. Sometimes the host rejects it, sometimes they takes it in, right? Even heard like they rejected the organ or wasn't compatible. And then that particular area, whether it's the bone or the skin or the blood, are monitored often to make sure that we don't have cancerous cells, because sometimes when we do those types of things, the DNA, like there's the regulation in the DNA and the growing is not correct, right? So we definitely do more follow up with those. Okay, here's two different specific examples of stem cell therapy. We will look at those in class. Parkinson's disease, probably the one that you've heard of the most, is when dopamine ceases to be made in your brain. Dopamine's in charge of lots of different things: schizophrenia, Parkinson's, too much, too little. Parkinson's is when you have too little. Those cells die off, right? Those cells that actually create the dopamine in your central nervous system die off. And so it's treated by replacing those dead cells with functioning ones that still create dopamine. Okay. Stargardt's disease is inherited from muscular degeneration in juveniles, so kids, and ends up leading to blindness. And again, it's treated by replacing dead cells in the retina, so the back of the eye, with functionings one to restore vision. Okay, here's a chart for you guys that kind of just goes over all those things that we went, versus the embryos, the type of stem cells, the embryos versus the umbilical cord versus the adult tissues. I think I misspoke earlier. I think I put those two together when I said the umbilical cord. So the embryo is within the like still growing, right? So while you've got a zygote, an embryo, and then a fetus, right? So an embryo is when it's still growing, right? So it's after a fertilized egg, in between, before it's an actual fetus. Those where you find the totipotent cells. So the umbilical cords are separate than those. Some people save the umbilical cord blood because it's easy to get out of there. But I know I said that earlier. I kind of mixed those two together. So make sure you have those as separate. This is the potency, right? So remember, embryos have the highest, they can become anything as they're creating different fetus. The umbilical cord and the adult have lower, but are still able to create many different things. And then the tumor risk. So with the embryos, we have the higher one because they are in that stage of just like multiplying, versus the umbilical cord and the adult tissues have much lower. Okay, this one's very costly. It's also controversial. Like I'm talking about why certain people can afford to do that and other people can't. So is it really an ethical decision? This one right here, the limitation about what they destroy, like it's talking about destroying an embryo or that it has already passed away or I don't want to say is still living, but about to not be, and very difficult to take out adult tissues. Okay. Okay. So this is a topic review of everything. We will go back over it in class and go through a lot more examples, especially with the magnification, because I know you guys love math, and we'll look at some different cool things with stem cells. You guys have a fabulous evening, and I'll talk to you soon.